Hello guys, welcome to my channel. We are studying about the chromosome. This is the part 2 of the chromosome. So far we studied chromosome function, like the structure of chromosome, how it becomes in the cell cycle, and now we are studying the structure, that how the chromosome binds the DNA. So this is the nucleosome, right? The basic packaging unit of the eukaryotic DNA that we studied, and the proteins that are responsible are histones and non-histones, right? The total 8 histones proteins, H2A, H2B, H3, and H4, like 2 of each, right? And this is the structure, right? So further moving in this topic, so like all four of these histones, this four of this histone that make up an octamer, right? So this four together make up an octamer together. Like the 8 proteins are relatively small protein. They are really small proteins with high proportion of positively charged amino acids. So this has a high positively charged amino acids, especially lysine and arginine. G9. So this is, I'm gonna write it down, positively charged. I think this is done. Where's the one? All right, so it has a very high proportion of positively charged amino acid. This is lysine and arginine, and what it does, this positive charge helps histones bind tightly to the negatively charged sugar phosphate backbone of the DNA. So as we study the structure of DNA, that the backbone of DNA is the sugar and the phosphate, and it is a negatively charged, and this is positively charged. So this is the reason it binds very perfectly with this histone proteins, right? And this numerous electrostatic interaction explain in part why the DNA of virtually any sequence can bind to the histone octamer. So any sequence of DNA can bind to this histone octamer. Each of the histone in octamer also has a long, unstructured N-terminal amino acid tail as well that extend out from the nucleosome. So for example, this is the nucleosome, and a tail comes out of the nucleosome, extends, and these histone tails are subject to several types of reversible covalent chemical modification that control many aspects of the chromatin structure. All right, so although the long strings of this nucleosome form, so these are the long strings of this nucleosome form on the most of the chromosomal DNA, but the chromatin in the living cell rarely adapts the this bead structure, rarely adapts the extended bead structure. In the living cell, chromatin does not have this bead structure. It rarely adapts this structure. Why? Because the nucleosome are further packed. So this is going in the cell cycle, this process, and once it is done like this, still it's not totally packed. So in order to pack, so the nucleosome are further packed on top of one another. So this is the bigger picture of this. So if this is like this, so the nucleosome will start to adapt like, or to pack like on top of each other to make a very compact structure, such as the chromatin fiber. Remember that I showed the picture that the chromosome, how it looks in the metaphase, the dense chromosome, that structure is formed after packing of this nucleosome on top of each other. So this additional packing of nucleosome in the chromatin fibers depend on the fifth histone, that is called histone H1. Should I do it here? So the packing of that is done by histone 1. So that is done by this histone 1, which is thought to be pulling the adjacent nucleosome together into a regular repeating array. So for example, this is our beads like structure, and what the histone 1 do, So histone 1 is something like this. This is our histone 1, right? So, and it has the carb terminal carbon and then amino carbon tail, and it has a tail. So the histone 1 consists of a globular region, plus a pair of long tails at its C terminal and N terminal. So N terminal and C terminal both has a long tail, right? So what it does, so for example, like this is one of the core nucleosome, and then not right and right. So this goes like this, and this goes and helps in packaging this nucleosome together over top of one another. So this is a kind of mechanism that is happening after the beats, and histone 1 is responsible for this. Right, so you can see a image on your screen. So that is the entire process how the DNA packing occurs on a several levels in chromosomes. So we have seen the short region of DNA double helix, right? Then we studied the beats on a string that I draw the picture that you can see more clearly now. And then, as I mentioned, the histone 1 is responsible for chromatin fiber of packet nucleosome, right? And after that, the chromatin fiber folded into loops and it gets more compact, and then you can see the entire mitotic chromosome, right? So this is the net result, and each DNA molecule it's written has been packed into a mitotic chromosome that is 10,000 fold shorter than its usual extended length, right? So this was the structure and how the chromosome is packed very compactly. So we studied the structure of chromosome and how the DNA is packed in the structure of chromosome. But the question arises that it is so densely packed as you saw in the picture. So when the cell needs the DNA, how it can be rapidly provided when the cell needs the sequence or a part of DNA. So which protein is responsible for that? I will show you. Maybe I will remove this section from here. Alright, so eukaryotic cells actually have several ways to adjust the local structure of chromatin rapidly. So there are several ways, but one way that take advantage is the protein known as chromatin remodeling complex. Chromatin remodeling complex. This is one of the protein. This protein machine uses the energy from the ATP hydrolysis to change the position of the DNA in this wrapped around the nucleosome. So how it does? So, for example, I'm not really good in this picture, but we'll try to show you. Maybe so. I'll show you. Something like this, and all right, I'll draw it from here, and we know it has our DNA that is wrapped around like this, something like this, right? It's not so much, but we'll make this, right? So this is our DNA that is wrapped. So, so there is this chromatin remodeling complex protein, and this is depend on the ATP hydrolysis. So it is known as an ATP dependent chromatin remodeling complex. So it shapes is something like this. Not sure if uh, Does that make sense? I'm not sure if that makes sense or not. But it's like a spin. So if this is a round and this chromatin remodeling complex has a shape like this, so it will turn and then we will have, so we'll do ATP to ADP, right? Oops, sorry, ADP, right? And then, so right now this is just like this. So after it will spin. So for example, here we have the DNA part that is required, and so on further, so this will go and maybe it will come more outside, right? If you have a 2 or 3 strands of DNA that are wrapped around this nucleosome core, so after chromatin remodeling, whichever part is needed, so it will act on that and like it will spin the nucleosome chromosome, and then you will have that part, right? So and then again we will have this that is acting on it. It's not a very good picture, but I hope you understand that. Or you can, or I'm posting a picture more clear that you will understand. But this picture on your screen, you can see this is how it is actually processed, something like this, right? All right. So the complex which attach to both the histone octamer. So this complex attaches both to the histone octamer, that is our nucleosome, and the DNA wrapped around it, can locally alter the arrangement of the nucleosome on the DNA, right? Making either more accessible or less accessible to other proteins in the cell. So during the mitosis, when mitosis is happening, many of the chromatin remodeling complex are inactivated. Why? Because this may help mitotic chromosomes maintain their highly packed structure. Because if this is available and they get uh, ATP that is hydrolyzed, so they will act on the packaging of the chromosome and it will become more loose instead of packing further, right? And so the another way of altering chromatin structure relies on a reversible chemical modification of the histones. So as we know, these histones have different tails. So the tails of all the four of the core histones are particularly subject to this covalent modification. So you can see the image. So this is how the histone looks, and it has all the H3A, H2A, H2B, H3, H4 tails it's shown. So for example, like acetyl, phosphate or methyl groups that can be added to and also can be removed from the tails of these histones by enzymes that reside in the nucleus. So there are enzymes that are present in the nucleus and that can add like these proteins, or that can remove these groups from the tails, right? So what happens, so this and other modification can have important consequence for the stability of chromatin fiber, right? So this is our chromatin, right? So the core nucleosome and DNA, everything. So for example, like if acetyl or methyl or phosphate group are added, so there will be modifications in the tail. So for example, like acetylation of the lysine, right? We studied there are lysines in this histones. So acetylation of lysine can reduce the affinity of the tails. Affinity of the tails, like this is our tail, right? A tail part of the, sorry, all right, this is our histone. So this is a tail part of our histone, right? So the acetylation of lysine can reduce the affinity of the tails for the adjacent nucleosome, thereby loosening chromatin structure, right? So if for example, if like here there is acetylation of lysine in this, so it will reduce the affinity of the tail for the next nucleosome and it will not bind tightly, instead it will become loose, right? So most importantly, however, this modification can serve as a docking sites on a histone tails. So for example, if this is a histone tails, right? If this is a histone tail, I will write it down here. Histone tail. For example, this is histone tail. And this all this modification like acetylation of lysine or whatever. So this serve as a docking site on the histone tail for a variety of regulatory protein, right? So if this is changed, so there will be a regulatory protein that can act on this. So it serves as a docking site, like the protein needs to dock. They will see if the modifications happen and the regulatory proteins are favorable. So this regulatory protein can dock on this histone tails, doing further modification. So different patterns of modification attracts different proteins. So, however, if an acetyl group is working or phosphate or methyl, whatever group is working, it will do a modification, right? Whatever modification, and depending on the modification, different proteins are attracted to the particular stretch of the chromatin, and some of this protein promote chromatin condensation. So, some of the protein that is attaching here promote chromatin condensation, whereas other decondense chromatin and facilitate access to DNA. Like the same. So this is how that's the that's the mechanism for the chromatin remodeling complex, and this is another kind of mechanism that is helping to condense or decondense the chromatin, allowing the access of the DNA, right? So specific combination of tail modification and the proteins that bind to have a different meaning for a cell, right? So for example, one pattern indicates that a particular stretch of chromatin has been newly replicated. Like for example, this attaches and like, so there is one example that it shows like, okay, this is part is newly replicated. Another indicates that genes in that stretch of chromatin should be expressed. Still other indicates that nearby genes should be silent. So these are different kind of expression that are happening when the proteins are attaching. So like the chromatin remodeling complexes, the enzymes that readily modify the histones tails are tightly regulated. Why they are tightly regulated? Because they are brought to a particular chromatin region by interaction with proteins that bind to specific sequence in the DNA, right? So the histone modifying enzymes work in concert with the chromatin remodeling complexes to condense or decondense stretches of the chromatin, allowing local chromatin structure to change rapidly according to the needs of the cell. So this is the different mechanisms that allows the DNA accessible or not accessible. Right, so now, so the localized alteration of chromatin packing by the remodeling complexes and histone modification has important effects on the large scale structure of interface chromosomes. So interface chromatin is not uniformly packed, right? So the interface chromatin is not uniformly packed. That means instead regions of chromosomes that contains genes that are being expressed are generally more extended, while those that contain silent genes are more condensed. So thus the detailed structure of an interface chromosome can differ from one cell type to the next, helping to determine which genes are expressed. So most cell types expresses about 20 to 30% of the gene they contain. Right, so you can see the image. So the structure of chromatin that varies along a single interface chromosome, right? So the most highly condensed form of interface chromatin is called an heterochromatin that you can see. So the most condensed is the heterochromatin. It was first observed in 1930, right, as a discrete strongly swaying region within the mass of chromatin. But the Heterochromatin typically makes up to 10% of an interphase chromosome, as you can see. And in mammalian chromosome, it is concentrated around the centromere region and in the telomeres at the ends of the chromosome. That's why you can see the heterochromatin is at the telomeres and in the centromeres. And the rest of the interphase, as you can see, chromatin is called euchromatin. So although we use the term euchromatin to refer to the chromatin that exists in a more decondensed state than the heterochromatin, right? So both euchromatin and heterochromatin are composed of a mixture of different chromatin structures, right? It has different chromatin structures as you saw. So one is more condensed and the euchromatin is less condensed. And each type of chromatin structure is established and maintained by different sets of histone tails modification that attract different sets of non-histone proteins, right? So as we studied, like the chromatins are made up of histone tail modification. So, and this histone tail attracts non-histone proteins, right? So this modification that direct the formation of most common type of heterochromatin, for example, include the methylation of lysine 9 in the histone H3. So that is complicated, but I will show you. All right, so I will show you what is happening, how the heterochromatin forms. Right. So, for example, uh, we have, I'm just drawing. Right? And maybe here, okay, this. I'm trying to make a very simple. Right, so for example, this is our chromatin. So just using as an example. So the first modification, what happens in that? So the modification that direct the formation of the heterochromatin is actually that is include methylation. So I will write it down. Here. I will write it down here. So that includes methylation of lysine 9 at the position 9 in histone H3 in histone H3, right? So this modification attracts the heterochromatin proteins. So for example, this methylation of lysine 9 in this histone H3, so it will start forming heterochromatin. So that means more condensed structure. I'll show you. Maybe something like this. And this becomes and this pink section that I have made, so that is a barrier DNA sequence. That's why I will write it down here. Barrier DNA sequence. So this is barrier, the pink one is the barrier DNA sequence, and what it's used for I will show you. So now this part is our heterochromatin and the entire thing, the other thing is euchromatin, that is not what condensed, right? So this methylation of 9 that start the our, this is just an example, that start the heterochromatin. So once this is established, once the heterochromatin is established, so it can spread to other nucleosome. Why? Because this histone modification, this tail modification that happens attract a set of histone chromatin or sorry heterochromatin specific proteins, and what it does, including histone modifying enzymes as well, which can create the same modification on the adjacent nucleosome. So once they start, so I'll show you. All right. When this, I'm just making the same structure here. To make it open. Right. So this once it has established, so it will attract, for example, a heterochromatin specific protein, right? And that also will attract some maybe the histone tail modification, right? So this will, what this will do is it will change the modification of the adjacent nucleosome as well, and it will start attracting over the line, right? And this modification in turn recruit more of the heterochromatin specific protein, causing a wave of condensed chromatin to propagate along the chromosome. So it will come, and then, for example, then we have more of this, right? More condensed structure, right? And we have just this left, right? So it will just become more condensed and condensed. So it goes until this heterochromatin will continue to spread all over here until it encounters this barrier. This is the barrier DNA sequence that stop the propagation. And in this manner, extended region of heterochromatin can be established along the DNA, right? So the DNA barrier comes, so it stopped here, and then we will have our euchromatin part, and then again, wherever another methylation of lysine 9 happens, and then heterochromatin can goes, and it goes until they meet the barrier DNA sequence, and it will stop. And so once we will have our heterochromatin and then euchromatin, heterochromatin and then euchromatin, right? So this is our interphase chromosome. All right, so most of the DNA that is permanently folded in this heterochromatin in the cell does not contain genes, because heterochromatin is so compact, the genes that accidentally become packaged into heterochromatin usually fail to be expressed. So such inappropriate packaging of genes in the heterochromatin can cause disease as well. So in humans, for example, the genes that encodes beta globin, so the genes that encode the beta globin, this beta globin, part of the oxygen carrying hemoglobin molecule, is situated next to the region of a heterochromatin. So if because of an inherited DNA deletion, so for example, during the inherited stage, if one of the DNA is deleted, that heterochromatin will spread, right? It will spread. The beta globin gene is poorly expressed, and the person will develop severe form of anemia, right? This is one of the example. Perhaps the most striking example of the use of this heterochromatin to keep genes shut down or silenced is found in the interphase X chromosomes of female mammals, right? In mammals, we know female cells contains two X chromosomes, whereas male contains one X and one Y. Because of a double dose of the product X chromosomes product would be lethal. So female mammals have evolved a mechanism for permanently shutting down one of shutting down or inactivating one of the two of X chromosomes in each cell. So at random, one or the other of the two X chromosomes condensed into heterochromatin early in the embryonic development. So thereafter, the condensed and an inactive state of the X chromosome is inherited in all of our many descendants of those cells, right? So you can see this is the picture, like, so one of the two X chromosomes, right? Each female cell contains X chromosome, one from male and one from female, paternal and maternal. So at an early stage in the embryonic development, you can see, so one of these two X chromosomes become condensed into heterochromatin, right? And each cell division, the same X chromosome become condensed and inactivated in all the descendants of that original cell. So thus all mammalian females end up as a mixture of cells bearing maternal or paternal inactivated X chromosomes. In most of their tissues and organs, about half the cells will be one type and the other half will be other type. Right? You can see, so one is active and one is inactive. Right? So when a cell divides, it generally passes on its histone modification. So whenever the cell division happens, it passes the histone modification, chromatin structure, and gene expression patterns to the two daughter cells. So such cell memory is critical for the establishment and maintenance of different cell types during the development of a complex multicellular organism, right? So this was the structure of chromosome and how the DNA is packed. Alright guys, thank you guys for watching and don't forget to subscribe.